I'd like to ask our morning speakers to return to the front and form a panel for answering questions from our audience. Dr. Lewis will serve as our moderator. Thank you all for the exciting work that you are doing and for this progress. The patients here today are but a few of hundreds of thousands around the world who hold out great hope for your results. Thank you again.
Nancy and Doris will be going around the room to pick up any questions you have, and we'll also give microphones to each of you if you want to make verbal questions. This is your chance to ask the experts. There are a lot of questions that come up, I'm sure, in your minds. A couple of questions came up in my mind. David, I have a question. You showed the treated animals through 10 or 11 weeks having actually gotten better than the normal muscle. Do you have a more extended follow-up on that after 10 or 11 weeks?
Okay, so well, a long wait for just answering no. So we haven't gone longer than that. On the other hand, AAV have been shown to be very stable, very capable of expressing the gene they carry for a very long time. There are studies in non-human primates, for example, reporting expression for at least 6 or 7 years. So AAV are kept in the, if the tissue is not actively replicating, like in a muscle in a steady state condition, they are kept for a very long time. So that's one of the reasons why they're currently tested for gene therapy approaches, because with a single delivery in the appropriate tissue, you will have the expression of the therapeutic molecule for years.
Thank you. Next question is for Dr. Emerson. Are you still looking for families to donate muscle samples, and if so, how is that tissue collected?
Yes, are we on here? Yes, we are still looking for families, and I think if you're interested, you should see Dr. Wagner, who sort of oversees this this process. We're particularly, as you saw when we began to look at a large number of families, there's significantly interesting, important families with unique genetics that have deletions of the locus that don't show disease pathology. So we're quite interested in that.
Are you interested in infantile disease?
Yes, and we've actually talked about the possibility of infantile. Another whole class of this disease is a possibility. So if you have an interest, please see Dr. Wagner. And she's here at the meeting.
Dr. Wagner is from Johns Hopkins in Baltimore, and she's in the, I guess you'll call that a green dress back there. So if anybody is interested in talking with her about making a contribution of muscle biopsy material, she's available and be glad to talk with you, I'm sure.
And she'll be speaking tomorrow in the panel, so that will be an important time to talk to her also and get her feedback. But she's like really an expert clinician. And there are various, so we are looking for different forms of the disease, like the infantile or early progression disease, which would be quite important for us to be able to sort out some of these issues. And so we do, however, feel that with the symptomatic class, we've done quite a bit of analysis that gives us confidence that we have enough families for looking at the traditional FSH1 to get statistical information, and through our work with biomarkers and sorting out family variables from the process. So that's a good question, and we are definitely want to keep doing this.
There are several questions having to do with vitamin supplements and possible nutritional aspects of the disease, and the questions are relating to such things as should patients be taking high protein drinks in the morning? Should they take certain supplementary vitamins, and what would they be? And what I suppose the panel at large might comment on this. Does anybody have any specific feelings about nutritional aspects?
So I'll say something about the vitamin C. I think that might have been directed at antioxidants. So I would say that it's data that you see in the laboratory. It's a long way from there to actually prescribing that in a patient, and so I'd be very, very careful. On the other hand, if you're like me and you have a typical Western diet, you don't eat enough vegetables and green things, you probably actually don't get a normal amount of antioxidants. And so like a normal level of supplementation is probably a good thing for almost everybody, certainly to eat your vegetables. But the first rule of pharmacology is the difference between a drug and a poison. I'm not sure if you've all heard this, but the main difference between a drug and a poison is the dose, right? And so anything that is taken to excess can be extremely deleterious, including antioxidants. So I'm kind of suspicious of some of the health sort of products that are promoted that give you extremely high levels of anything. I think those are all probably in some people can be deleterious. Vitamin C can contribute to kidney stones if you're predisposed to such a thing. And so, short answer is vitamin supplements probably aren't a bad thing, but I certainly wouldn't overdo it, and certainly not based yet on the work that we're doing.
I'll make a comment. With our Wellstone repository collection in our cells, we have published a paper recently on whether FSH muscle cells are more sensitive to oxidative stress, which has been in the literature from small, unstatistically powered studies saying that there was an issue with that they might be, FSH cells might be more susceptible genetically. So we did this study, which is published, and found that families vary in oxidative stress response, but it's not correlated with disease. And again, it shows the importance of looking at a lot of samples, particularly that families carry certain inherited characteristics like you might expect, like oxidative stress susceptibility is one of them, but it's not related to disease. So when you look at a large number of families and subtract out the genetic component of family versus disease, we saw no difference in the sensitivity of FSH cells compared to from unaffected cells in oxidative stress. So, again, it's that's just a fact. So,
I think this is probably for Dr. Kaiber. The reference was made to the fact that you get so many contractions in a muscle before the muscle will become disordered. And does that imply that you should not over-stress a muscle in terms of exercise and physical therapy and that kind of thing?
I think that's a really interesting question, actually, and we really don't know. So certainly you could make the argument that if your muscle has less of an ability to regenerate, then actually damaging it through excessive overuse, as we do if we lift weights or participate in a sport that uses certain muscles particularly aggressively so that they overgrow and hypertrophy, that might actually be deleterious. On the other hand, we also know that not using our muscles introduces problems as well. So muscles will tend to get sick if they're not used. It is really important just sort of for overall well-being, not just of muscle, but other cells in the body that you get some kind of regular exercise. And so, but I think it really has to be tested, and in fact, it's a very, very difficult test to do in a disease like FSHD, where the symptoms are slowly progressing, is exercise over and above the normal level of exercise. Is that a good thing or a bad thing? I think we just can't say. And I think the best course would just to be cautious, to not be afraid of exercising, but at the same time to not look at it as a potential remedy, and certainly not to overdo it. But obviously there isn't any strong data to support that either direction.
Thank you. I think maybe Dr. Tapscott could answer this one. It has to do with the question about whether there are differences in family members who have the genotype but not the phenotype of FSHD.
So if I understand correctly, the question is the penetrance of
Right.
FSHD in different
That's what
families.
I would infer.
And so, as was brought up by Dr. Emerson, there are individuals that have either the deletion of the repeats or the lack of the chromatin structure of the repeats that have not gotten FSHD. And I think there are two things there that are going to be very interesting, and if we have this meeting again 2 years from now, I hope we have the answers to them, is the FSHD2 families are really telling us that there are significant modifier loci in the population that alter the penetrance of the disease. And once we identify those genes, we'll be able to say whether they also contribute to the penetrance of the FSHD1 phenotype in individuals that have the contraction, because if there are these modifier loci segregating in the population, they could modify both FSHD2 and FSHD1. And so that's kind of a long answer to saying that this incomplete penetrance can be exploited for research purposes, and I think will eventually, and probably fairly soon, lead to a better understanding of the pathways that result in the heterochromatization, or as I was saying before, that sending the gene to the attic. And so that once we use those people who don't have the full phenotype to identify those genes, we'll be able to have another tool in our arsenal for targeting the pathways for drug development.
Thank you. And this is for Dr. Gabellini. The questioner says I missed your point on whether FRG1 is one of the genes within the human D4Z4 region, and maybe expand more on what the implications are as far as FRG1 has to do with FSH.
So yes, so FRG1 is located in the FSHD region, and it was originally reported by us as being part of among the genes that are whose expression is increased in FSHD patients. Because of this, and because of the fact that we also made mice that express an increased level of FRG1 that develop a muscular dystrophy, we initially were supporting an idea that if the aberrant expression of FRG1 was important for FSHD. On the other hand, the problem with FRG1 is the fact that a number of other groups actually failed to confirm its aberrant expression in the patients, while later on, the aberrant expression of DAX4 was reported, and as you have heard, it is much easier to confirm DAX4 overexpression in FSHD patients. Because of this, right now the leading candidate gene for the disease is DAX4. FRG1 may contribute in the disease, may even be one of these modifier genes that Dr. Tapscott was referring to. This would be actually one that contribute to worsen the phenotype, but let's say that the data that we're getting from the patients at the moment suggests that DAX4 is the best candidate for the disease.
Thank you. There's one for Dr. Emerson. It says that you use the FL symbol in your slide about DUX4, and what is the FL?
So DUX4 is a gene, and Stephen Dr. Tapscott can speak to this as an expert, but the gene itself, because genes are, when they produce their RNAs, are spliced from exons, you get alternative forms of the RNA, some which are shorter or longer, have more coding or non-coding sequence. So each exon, there's a chemistry involved in taking the primary transcript from the gene and then sewing it back together as a smaller transcript. DUX4 gene has at least 4 different transcript sizes because of this splicing, and the long form is the form that produces the DUX4 protein that's toxic, and that's what the FL is, full length.
Another question for you has to do with the question about humanizing mice, and how does this help, and what more, maybe for general consumption, what would a humanized mouse look like?
No head. So the idea is you take a specific muscle, like in the case of the TA muscle, the tibialis anterior, and remove it or destroy it, but leave the scaffold there. You can add stem cells back to it, like from the repository, and they will go into the scaffold of this destroyed muscle and rebuild it anatomically. And that's what. And Dr. Wagner has a variant, important variant, where she takes the whole muscle out from the mouse and replaces it with big chunks of human muscle taken out from surgical biopsies. And this allows the muscle to actually rebuild, and so you get an anatomical muscle that gets innervated, and I didn't talk about that, by mouse neurons, but and so it actually forms an in vivo contractile muscle. So giving you an opportunity to study the pathology of the disease in a context of an animal. So this is important for a couple of reasons. One, in cell culture, where we do a lot of our work, you only can go to there's no innervation. It's a very primitive, just muscle system, which can show you the types of genes that can be disease genes in a simple way. But in the animal, there's the complexity of muscle contraction and muscle physiology. So it gives you a chance to study that. And also, if you're trying to develop a drug, developing a drug in tissue culture is one thing, but being able to deliver a drug into people requires understanding how that drug is metabolized in an animal or before it goes into people. And so having a disease model in a mouse is particularly important to develop a drug and part of the whole preclinical testing scheme. What is cool about this is that it makes no assumption, a humanized mouse makes no assumption about what the bad gene is, whether it's DUX4 or FRG1 or whatever. You're putting the diseased muscle stem cells in and letting them rebuild a diseased muscle in an animal. So if you have biomarkers for the disease, you can use that as a way to monitor this, independent of what the primary cause of the disease is. And you can develop treatments that way. So that's the advantage of this model over using a selected approach where you say DUX4 is the bad guy, let's make a mouse that overexpresses DUX4, which already assumes that DUX4 is the bad guy. So it's just an alternative. There's value to both approaches. So is that, yeah.
Thank you. Dr. Gabellini, the question is, does DUX4 siRNA treatment affect muscle cell cultures of FSHD1 patients to promote muscle fiber development?
So this does not, to the best of my knowledge, but maybe Dr. Tapscott want to add more, tested yet. There's been a report of a group that use mouse to test this, to address this, and what they did, they used two different viruses. One that was expressing high level of DAX4, and the other that was expressing this small RNA molecule, siRNA, against DAX4. And so if you inject only the virus expressing DAX4, you basically destroy the muscles. This gives a very severe phenotype. If you co-inject the DAX4 virus and the DAX4 siRNA, shRNA virus, you prevent this phenotype. The issue here is that this is a very artificial system, because you are co-treating, so it's very far away from the situation in which you will have a patient that have already developed symptom of muscular dystrophy, which would be most likely the therapeutic setting in which we will have to intervene, because in the future, once we will have drugs that we would like to test in patients, most likely the setting will be individuals that have already developed sign of muscular dystrophy, because otherwise before they will never arrive, most likely not arrive at the attention of the clinicians. So you will have individuals that have already developed muscular dystrophy, and you want to intervene either to slow down the progression of the disease, or to even rescue and rebuild muscle in order to ameliorate the symptom of these patients. So the approach that has been used so far is very far away from this setting, because you take animals that have nothing, you inject one virus or two viruses at the same time, and this doesn't really bear much significance to a possible future therapeutic setting. But maybe Dr. Tapscott want to add something.
Dr. Tapscott, can you explain why statins have an effect on muscles, and does that have anything to do with FSH?
No, I can't.
Statins, as you may know, are the medicines that are used for lowering LDL, the bad kind of cholesterol, which in some cases leads to premature arteriosclerotic disease. And what happens is that in some patients, certain of the statins may cause a muscle aching. I'm not aware of any actual atrophy or dystrophy that would come from that, but it's uncomfortable. Maybe you can talk to Dr. Wagner later. She can. You have any other comments about that, Catherine?
Patients, it's not a muscular dystrophy, but it is a toxic myopathy. And if you have alternatives, I usually recommend people trying alternatives to lowering their cholesterol rather than statins. It's not that people with muscular dystrophy are more at risk to developing a toxic myopathy, but it's harder to tell. You already have some weakness, you already have an elevated CK, and we might subscribe we might think that it's just the progression of the disease when in fact you have a superimposed toxic myopathy. So that's really the key then. And there are other ways. There's a drug called Zetia that decreases the absorption of cholesterols from the gut, and it is not known yet to cause a toxic myopathy. So that's one good alternative.
Thank you. Next question, are any of you accepting internships, and when can I come?
Definitely.
Is the panel in agreement that DUX4 is the candidate gene, and with the DUX4 target mouse model possibly lead to faster results? Dr. Emerson, maybe you can answer that.
I think the data that DUX4 is an important player in disease is very substantial. I guess the question is And I think, it's, if you had to, clearly, I mean, our work sort of addresses the question of whether it's sufficient. It may well be necessary, but is it sufficient? What we don't really know is, and we know that DUX4, in a sort of an unbiased screen of FSH-affected cells, the target genes are activated, which is further evidence that DUX4 is not just expressed but has activity in FSH cells. But it also has activity in cells where there's no pathology and in muscle tissues that don't have any pathology. So again, having a necessary but not sufficient gene, it's very important to keep working on it and developing the kind of mouse models that can test what we don't know is what is the toxicity, the results that in muscle loss in response to DUX4 or whatever the gene is. What is the pathology all about? And how do you connect that up with this? And Stephen talked about an interesting idea of misexpressing some genes like defensins that might be responsible for the pathology. But where the gap is where the pathology is, and can we reconnect it back to DUX4, and then can we connect whatever keeps DUX4 modulating its function, can we connect that into the system now? So we have probably DUX4, we have modulators maybe of DUX4 function, we need to connect to pathology. But I think Stephen should address this some more. Do you want to? Yeah.
Yeah, I think it was kind of directed at the whole panel, so I've got a comment as well. And just to actually go back one question to anyone who's interested in participating in research, if you have some particular skill that you think is applicable, then I think we probably all are open to interacting with patients who think that they have an ability to contribute, whether it's on the bench or with computer skills, et cetera. There are ways of interacting, I think. And so certainly in my case, just send me an email if you'd like to interact with the work that we're doing. On the question of does the panel agree that DUX4 is the gene and the DUX4 mouse is the way to go, as you've heard, there's some consensus on the panel, but there will always be, for the next several years, I'm sure there will be discussion and argument at scientific meetings about what is the real gene. You shouldn't, from my perspective, you shouldn't let that stop you from doing the next experiment, because if you let that stop you, then the whole progress of march toward a cure for this disease will just be delayed. And so, to some extent, each of us has taken a risk. Certainly, I can say that when we decided to invest deeply in DUX4, it certainly wasn't considered a very interesting gene for FSHD, and we had our own reasons to think it was interesting. We took a big risk. And everyone takes that risk, and sometimes it can be quite difficult to get funding if you can't convince the people that are your peers that this is the thing that we should all be studying. And unfortunately for FSH research, I think that has limited the progress in the field over the last decade or more. And you'll see, and talking to many people, I know many of you share the same frustration, which is that muscular dystrophy foundations, the MDA, et cetera, tend to focus on certain diseases, and FSH often gets left out. And that's because there's really good arguments for what you should be studying in Duchenne's, and everybody's in discord about what we should be studying in FSH. And so if you take the approach that you have to wait until everyone's in agreement, then we won't make any progress. And so I think it's okay for there to be some dissent. I think it's healthy.
So I'll just quickly add to that. First, I think it's admirable if anyone wants to go into medical research, particularly FSH research, and training people and getting people involved is something we all do, and so that's important to say. Secondly, I want to echo what Drs. Emerson and Kaiba said. Is first, I think the genetic and molecular evidence is overwhelming that DUX4 is a major player in the cause of FSHD. However, our job as scientists is to wake up every morning and say, what could we have missed? Do we have this right? And so there's going to be a lot of noise about, what else could it be, modifier loci, this, that, the other thing. And it's important for us to keep ourselves honest as we go through this. But I do want to emphasize that I think that between 2 years from now and now, we can coalesce around a real dominant model for FSHD, and that's important for the reasons Dr. Kaiba said. is it's very hard for NIH or other agencies to step forward and fund research unless there's a consensus within the scientific community about what the right thing is to do. And I think we're really coming down towards this consensus that focusing on the role of DUX4 and what the specific mechanisms are, what the modifier loci are that cause FSHD. And so we should be able to really accelerate progress.
Thank you. Dr. Emerson, can you explain the variable expression of muscle damage in individuals and why it expresses itself later in life? And are there any protective mechanisms present early in life?
Well, I don't have an answer to that question. I mean, what I think we were, in our study, we were very surprised in our finding individuals who show all the genetic hallmarks of this disease without any pathology. And those individuals are very important to study and understand. It was also a great surprise that those individuals expressed DUX4 in both their biopsied muscle as well as in cell culture stem cell models that we have. Again, this is connecting the pathology of the disease to these candidate genes is really important, and at the molecular level. And so I think this is all just emerging now. Now that we've had this really important candidate gene like DUX4, which is the genetic and molecular evidence that it is a player is very strong, but having these individuals that escape is extremely important for understanding what the disease is and how we might treat it. This is not unusual. For other diseases, there are modifiers. This is not of disease progression, and this is not something just for FSH. This is a way, but different kinds of diseases have different kinds of modifications. And so, people with breast cancer genes they don't always get breast cancer, even though they have the genetic basis for it. So it's the same here. And so understanding that is, as I tried to make my point, was that is a way to think about how to treat the disease. If there's escapers or if there's this kind of genetic variability of penetrance, then it's very important as we develop therapeutics. And I think we'll be able to tease that out there. The tools are there now. We've got the genetic tools to look for modifier genes with the genome biology that's now available, the human genome biology, and we've got ways to look at gene expression that can ask whether a person that may be expressing DUX4 expresses a unique set of genes that are related to protection. And that's why having this kind of variation in our analysis is important. So,
thank you. Dr. Kaiber, the question has to do with doxycycline, and from a clinical aspect, is it wise for patients with FSHD to avoid doxycycline, or are there other agents that they should specifically avoid?
So no, there's nothing wrong with doxycycline. It's we use doxycycline in the laboratory because we've designed a genetic system to turn DUX4 on if we give the cells doxycycline. But if you haven't modified the cells so that they have a sensor for doxycycline, we actually take a protein from bacteria that is resistant to doxycycline because it can sense, it has a protein that senses the drug, and then that protein turns on genes. So we take that protein from bacteria, stick it in the cells. Now the cells can do what the bacteria do, is respond to doxycycline, turn on the gene. That's completely artificial and unrelated to why DUX4 may come on in patients with FSHD. So there's absolutely nothing wrong with doxycycline. It's just a system that we use in the laboratory.
Thank you. Dr. Tapscott, why does FSH affect certain muscle groups such as face and humeral muscles and scapula, but not the hands or feet?
Yeah, no, that's an excellent question. I think that FSH eventually affects most of the muscles, and it is most penetrant and earliest on in the upper extremity muscles. And I was trying at the end of my talk to give at least an evolutionary speculation as to why that might be. It might have been that the DUX4 gene was selected to leak a little bit in those areas. But I do want to say that I think that the work done by Dr. Emerson and others comparing affected and unaffected muscles, upper extremity, lower extremity muscles, will allow us to get at the molecular correlates of the differences. And as we get at the molecular correlates, we'll be able to go back and try to ask the why questions.
It's a very interesting question because, and this is what in our strategy, looking at like deltoid and bicep, is it that in your body that different muscle groups have different protective mechanisms against DUX4? That would be another way to look at it. The same issue is whether different individuals, but within your body, some muscles maybe have these kind of protective mechanisms in it. And we know that muscle gene expression, anatomy, all this, they're quite different. And so that's a way to look at the same question of modifiers, which is important. Yeah.
Thank you. Dr. Tapscott, does FSH affect only skeletal muscles, or may it also affect smooth muscles such as cardiac or other smooth muscle groups?
There are definitely other tissues affected by FSH. For example, there's a retinal vasculopathy in some individuals, hearing loss in some, and whether that's due to the same process as the skeletal muscle disease, I think still remains to be determined, and not much has been done there since most of the work has been focusing on skeletal muscle. So yes, other organs are likely to be involved, but the clinical phenotype is predominantly skeletal muscle.
Thank you. Dr. Kaiber, with an inflammatory component in the disease, are there certain anti-inflammatory agents that patients should be taking?
I think Dr. Tapscott might have a deeper comment on that. But so far, the things that we've discovered that interfere with DUX4 aren't anti-inflammatory. And whether or not you should be taking anti-inflammatories is kind of a question for your clinician, really, not a question that I think the work that we do has much bearing on. Do you want to contribute?
I think that's, again, a good area to think about over the next couple of years. And as I put forward in the talk that I gave, it's quite possible that an immune-mediated response is a component of this. And getting back to the earlier penetrance questions, everyone's HLA type and ability to mount an immune response is different. And that would be a very attractive way of explaining some of the differences in penetration, as the differences in the immune repertoire that every individual has. However, that's a model of the disease now, and we really need to investigate a little bit more directly whether that's a dominant component of the disease process. And the good news is we know what to do, and 2 years from now we'll be able to give you, I think, a pretty good answer.
One of the questions regarding inflammation was specifically, should you be taking low-dose NSAIDs on a daily basis? And
well, I take low-dose NSAIDs on a daily basis, and I don't have exposure to an FSHD gene. And so for FSHD, there's no evidence at this point. For other medical reasons, there might be reason to do it.
One of the questions has to do also with how do scientists communicate with each other so that everyone is up to date on their latest developments in the
Carefully.
field. And also how freely do you give up your own personal philosophies or insights into the disease? Any of the panel members can answer that.
Well, I think science is based on the exchange of information, and the meetings such as this, scientific meetings, we get up and we describe what we're doing and why we're doing it, what our theories are, how we're going to test them, and we open ourselves to the noise that I was talking about before of people questioning what's happening. And so there's I think I've been to 3 different meetings this month to present our work on FSHD as well as other projects to other scientific groups, well before publication, and then finally when things are published, it's an opportunity for people to see the full scope of the work with all of the supporting material, and to quite honestly criticize it and try to find the holes in it. And so there's a lot of argument in science, and it should be that way, and we're all in it to try to find the truth.
I would just add to what Stephen said, which is that we have the FSH Society supports a very important meeting, the FSH Workshop, which meets annually. It's met in Boston the last 2 or 3 years. It's going to meet in San Francisco with the Human Genetics Group again this year, which just brings FSH researchers together to really hammer out what the latest findings are and argue and try to look at data critically. And these are very important meetings, and the Society plays a key role in promoting this kind of interaction. Science is all about communication and communication of ideas and challenging ideas, and so face-to-face is email does not do it. Face-to-face meetings are very important to really be able to hammer out the complexities of what we're trying to do. And so I thank the Society for supporting one very important meeting for FSH research, the workshop.
This one's labeled a curiosity question, and I suppose it could be for all the panel members. Why did each of you become interested in FSHD research? And also the comment is, thank you for your efforts on behalf of families and patients. We are so grateful. Anyone have a comment about it?
I can say I'm a neurologist, and I've been doing neuromuscular neurogenetics clinics and coming across individuals with different muscular dystrophies, myotonic, FSH, and so one of the real reasons is the personal motivation to do something for these individuals. FSH was particularly attractive from a research point of view because the mechanism of the disease when we entered the field was so unknown, and it was a real mystery. And so as a scientist, at least I'm drawn to the difficult problems, the ones that haven't been answered, where one has the potential to make a real difference. And so FSH was a real challenge in that regard.
So in my own case, my background is as essentially a stem cell biologist, and I had a kind of peripheral interest in stem cells of the muscle and how they work. Mostly what I was studying at the time, and still study to some degree, are the stem cells of the blood system. And I became interested in FSHD really through, there's kind of 2 parts. One is an interaction with an individual with FSHD who got me interested in looking at this disease and finding out more. And then the second was just looking at the genes that are present in the locus and discovering that one of these genes has an apparent connection to a stem cell gene of muscle. And from that connection, it seemed to me that it was a really interesting opportunity to test that hypothesis. And I should say that none of the early work that we did anyway was funded by the Muscular Dystrophy Association or the FSH Society or the NIH. And in my own case, I was very lucky that the institution that I work for had a donor who was very interested in FSH research and who provided seed funds to do some of the early experiments that generated the data that are now funded. And so it was quite a set of different circumstances that all came together at the same time in my case, kind of just a lucky confluence.
So in my case, my background, so I'm not a clinician, I'm not a medical doctor, I'm a biologist, a molecular biologist. I always studied mechanisms that control gene expression. And the connection with FSHD was through a clinician that was studying the disease and that described all the fascinating aspect of this disease. It is this. I'm going back to what Steve has said. So, from the scientific point of view, FSHD is a very challenging but also very fascinating disease that I'm convinced that is through the study of FSHD we can learn important lessons about how regulation of gene expression is taking place in human being. And then as a scientist, if you have to pick a project, you're better off, or is more stimulating to pick a project that not only is interesting and fascinating, but has relevance for human health, whereby if you are doing good work, you may end up also affecting in a positive way other people's life. So in my case, again, it was a sort of serendipitous. Unlike Michael, I was supported by the FSH Society from the start. So my first fellowship, postdoctoral fellowship, when I started working in US on FSHD was from the FSH Society, which provided for my salary, and this continued through a support from the Muscular Dystrophy Association. So I've been throughout my history of working with this disease. I've been supported by FSH, the FSH Society and the Muscular Dystrophy Association, which really allow me to do what I did.
I became involved in FSH research about 5 years ago for two reasons. One, Dan Perez, and the second, my daughter. I was at Penn School of Medicine for a long time and moved to Boston to a research institute where I'm the director. And for a specific reason is that I wanted to be at a less medieval research setting than medical schools are, which are compartmentalized into departments and old-fashioned ways to think about things, and to do something really interesting, more collaborative, more multidisciplinary. As soon as, and the Institute had a long history of basic muscle research, which is my field. And so as soon as I walked in the door and sat down at my desk, Dan Perez called me up. He said, why aren't you doing something meaningful, which is treat my disease, find out something. And I've always been a basic science guy, and I'm trying to figure out how muscle genes work and how muscles are created. But I've always, when I went to Boston, I wanted to do something interesting and a little bit different, work on a disease mechanism, do something meaningful, as Dan said. So he kept calling me up. I didn't know the guy, and I didn't know anything about FSH either. And so he eventually just began to educate me and introduced me to some of the scientists who were interested in this field. And so then we decided to put together this Wellstone Center in our institute, which has this more flexible, collaborative idea model, and it was a perfect place to do that. Personal motivation is my daughter was born with brain damage, and she affects her muscles. She's in a wheelchair. She's grown up in a wheelchair. She's that's been her life, and I raised her that way. And I'm a family caretaker of somebody with a disability, and I've always wanted to do something that's sort of meaningful for people that are disabled and their muscles don't work right. And so I said, this is the way to do it. And I saw the disease as one in which we can make progress, and I think we are going to make progress, the kind of progress that's going to lead to really therapies.
I'll include myself in that group and say that I'm interested in FSH primarily because my wife and 3 of my 4 children are affected by the disease. And when my daughter was involved in a support group, I guess about 15 years ago, and that group which included Dan Perez, was the instigating force that drove for the development of the FSH Society. And my daughter asked me if I'd be interested in becoming a member of the board, and that's how I got started and how I've continued on since that time. And then that ties into the next question. I'm a neurosurgeon, and I have done muscle biopsies, so, and I don't know whether any of you have, but I can answer this question. What is the procedure for collecting muscle tissue? Is there pain? What's the anesthesia and the recovery time? The answer is that I suppose it depends on who's doing the procedure, but it's done with a local anesthetic. There's a small skin incision, and the amount of muscle that's taken out has to some extent, depend on what muscle is left. And you would want to have people who are not so totally affected, the muscle has disappeared. But the idea is to take out a big enough piece that, and Catherine Wagner can answer the specifics about how her lab wants you to prepare, to be the specimen to be prepared. But this recovery period is very relatively short. It just takes the time it heals for the skin to heal up. And that usually is a matter of a few days. If you want to get more details, you can talk with Dr. Wagner, who can give you the process by which her lab makes the specimens. And Dr. Emerson?
How many people in the room have participated in muscle biopsy? So those are people to talk to also.
Yeah, that's a good idea. That's probably the best reference you'll get. Next question is dealing with an article in the New York Times, which I didn't read. It has to do with junk genes. Is anyone on the panel aware of what this article is about? You might comment on that. One of the questions is, how does this tie in with cancer research and with FSH?
Those are great questions. That article came out after we published a paper in Science describing, or really providing, the genetic evidence that indicated that DUX4 was the most likely cause of FSHD. And at that time, it wasn't known whether DUX4, it was thought to be never expressed in normal human tissues. And we had not yet published the work showing that it is really normally expressed in the germline. So at the time the article was written, it was felt that it was a retrogene, which usually becomes pseudogenes that are the junk portion of the DNA that had no normal role. Subsequently, we've shown it's not junk at all, but it's a real gene and it really has a normal function. I'm sorry, the second part of the question was of cancer.
Yeah, is there any relationship to cancer research?
It actually, so I work at a cancer center, and I've worked on muscular dystrophies there for a long time, and I'm very glad to say that finally there's a link between this and cancer. And that has to do with the cancer testis antigen angle to it. If there is an immune response against these proteins being re-expressed in skeletal muscle, these germline proteins being re-expressed in skeletal muscle, that is probably exactly the same process by which cancers express these germline genes and get an immune response against cancers. And so the Cancer Institute now is very interested in looking at this relationship, and I'm getting a lot of cancer researchers interested in looking at FSHD in terms of its immune response, because if it's the same thing, we could leverage this for a vaccine against cancers as well as a treatment for FSHD, and so it's generating a lot of interest in the cancer research area.
Thank you. There's a question regarding fundraising and what's referred to as microfunding from the public. I'll answer that by saying if you're interested in fundraising, the FSH Watch and other communications from the society can help you to develop any kind of fundraising project that you'd like to become involved in. And as Judy can testify to, it's possible to have some pretty massive amounts of money that can accumulate from local efforts. She and her team have, as I mentioned earlier, raised several hundred thousands of dollars for FSH. And that I might add is our most, at this point at least, most productive single project. But we'll certainly eager to help with anybody who's interested in any other fundraising.
Excuse me.
This is a complicated...
Hello, sir. I'm sorry to interrupt. Sure. I was the one who asked that question because in the creative community there are websites like Kickstarter. Is anybody familiar with Kickstarter.com? It's like where young, and sometimes not so young, filmmakers or acting companies or whatever go to raise money on the web. So they basically just go there and list their project, and they get $1, $10, $25, but from thousands of sources, kind of like what Obama did. Some people were happy about it, some people not so much. Where? Okay. So I'm wondering if that exists for the scientific community on a project-by-project basis, and it might be helpful.
Thank you. Jim Chin is one of our board members, and he's going to be talking about development and projects also.
Right, we'll be talking about development a little bit later today, but also we're having a workshop tomorrow afternoon, and we'd welcome you to come and tell us about that. We haven't gone down that road yet, but we're open to all good ideas, okay? So thank you. Please come tomorrow, and let's talk about it.
Could I just say one thing about development, too?
Sure.
In the fundraising we've done, we've had tremendous backup from the FSH Society. Nancy has been there 100%, and whoever will be our new executive director, I'm sure will also be there for you, but could not have done this without the support of the society.
Dr. Emerson?
I can't emphasize how important the funds are that the FSH Society have provided for laboratories around the world to do FSH research, including our own group. And it's just really important. And as Stephen was saying earlier, you know, when a field which is emerging like ours, having private funding is really important because government funding supports things that are going to work. And here we're in the field of figuring out what's going to work. And so it's really important source of funds. And I hope you would feel fine in asking me, and I'm sure any member of the scientific community, to be partners with you in fundraising, to articulate what we're doing. Can be very important, powerful message to people that really can communicate the promise that research has. And so I think scientists can be very important in that whole process.
And I just want to add one thing to the comment about funding for FSHD research, is that there's really a lot of money. This country spends a lot of money on basic research. And in addition to the government spending all that money, there's the MDA, and mainly the MDA. And as I discussed a little earlier, that historically FSH has been underfunded. However, the people who make these funding decisions, they're very sensitive to feedback and criticism from, especially from affected individuals. And so it's actually really important that the patient community interact with the funding agencies on that level. Find out from the MDA how much of their funds are going towards this disease. And then find out from the NIH through your representative or whoever. This is something that Dan Perez is very, very good at, and actually that's one of the biggest contributions I think that he's made, is his ability to impact funding decisions that trickle down through the NIH to get a little bit more money from their billions sent towards FSH research. So if you feel that you can't contribute, you don't have a lot of, personally you're not wealthy enough to be a donor, and you can contribute in terms of helping with fundraising efforts, but you can also contribute, and I would say it's a very strong contribution, to interact with the MDA, to talk to their local people, and just get get them feeling the sense of frustration that we all have, that there should be more funding for FSH research.
Thank you. It's time to break for lunch. I'll just make a couple of comments. One is that, as Dr. Bellini noted, his career in FSH research began with a grant from the FSH Society. And there are a number of other researchers around the country who've had similar experiences. The Society does everything we can to promote independent workers developing an interest in FSH. A follow-up also on the MDA. Sometimes there's a little confusion among the general public about what the MDA does. Recently they've been more generous with FSH than they have in the past. Their primary interest has always been in what are thought of as Jerry's kids, and those are children with Duchenne's muscular dystrophy. And I've had experiences myself with people who said, well, I give to MDA, and they assume that money gets filtered down to FSH. Only a small, very small amount of the money that goes to MDA ends up in our fundraising efforts. So if you're primarily interested in fundraising, the way to do it is through the FSH Society rather than through MDA, I would say.